Welcome to the plenary session for this afternoon. Hope you all had a good lunch. My name is Anil Seth from the University of Sussex, and it's a great pleasure to be chairing this session. It's on the neural correlates of consciousness, when, where, when, and how. And we have three terrific speakers this afternoon. We have George Mashour from the University of Michigan, we have Yan Zhu from the University of Pittsburgh, and Marine Vernet from NIMH in Bethesda. So George needs very little introduction. He's one of the co-organizers of this meeting, has been for several years, and it's a great pleasure to introduce George Mashour, who's going to talk about the neural correlates of consciousness. Thank you, George.
Thank you. Good afternoon. Thank you for coming. I'm going to be speaking about the neural correlates of consciousness, and I'll be doing so in a very focused way. So this phrase, this concept, this research program is very important. It's been around for a little more than 20 years, but despite the great progress in the science of consciousness, despite the remarkable technical and experimental advances in neuroscience, there's still challenges in identifying the neural correlates of consciousness. In fact, there is still active debate as to whether or not the neural correlates of consciousness are, in very coarse terms, in the front of the brain or in the back of the brain. Now, you think that we would be at the phase of really homing in on the specific neural circuits or neuronal subpopulations that are mediating conscious experience, but even within the past 6 months, there was a debate in the Journal of Neuroscience regarding the question, are the neural correlates of consciousness in the front of the brain or the back of the brain? Now, you might note that I added on the neural correlates of anesthesia, and I've done so for two reasons. From the scientific perspective, I think there are a lot of interesting data about anesthetic-induced unconsciousness that can speak to this discourse. From the clinical perspective, we deliver more than 40 million general anesthetics each year in North America alone. And yet we have no standard monitor for the brain. We've had standard monitors for the cardiovascular system and respiratory system since the 1980s, but we don't have a standard monitor for the brain, despite the fact that the brain is the target organ for our most potent general anesthetics. The reason why we don't have a monitor is because we don't yet know where or what to measure. So, in very simplistic terms, proponents of integrated information theory, Drs. Tononi and Koch, suggest that the correlates of consciousness are in the posterior cortex, the so-called posterior cortical hot zone. They're advocates for prefrontal correlates from higher order theory and also the global neuronal workspace theory. Not to suggest that prefrontal activity is sufficient per se, but a critical player in the neural correlates of consciousness. I'd like to note that I do not have strong biases. I'm not here with an agenda. I've drawn on both of these theoretical constructs, but I do want to follow this line of investigation, suggesting that the neural correlates of consciousness and also of anesthesia relate to the neuronal workspace and long-range connectivity. This was an article that was published in 2007. I was a first-year faculty member at the time, suggesting that visual perception, visual consciousness was not correlated with early activity, both in terms of space in V1 or even higher order visual cortex, or early activity in time in terms of shorter latency evoked potentials, but rather was correlated with the activation of a wider network that prominently involves the prefrontal cortex and that is associated with longer latency potentials. Now, it's important for the purposes of this talk to point out that not only is there processing in the feed-forward direction from V1 to the prefrontal cortex, but there's also feedback processing. And this appears under a variety of different guises in the literature. It's been termed reentrant processing, recurrent processing, reverberant processing, reafferent processing, feedback processing. The idea is the same. Given a hierarchical brain organization, higher order areas receive input, but then they also feed back and influence more primary or lower order areas. Now, lest you think that I'm stuck in the literature from 2007, I want to point out that there was a recent article, it was alluded to in the opening plenary by Professor Mashour, published in Science in the non-human primate brain with very similar conclusions, that unconscious processing was associated primarily with feed-forward activity, but consciousness was associated with a wider spread network that was ignited, if you will, by activity in the prefrontal cortex. And you can see the model down here where you have both feed-forward processing, you have local activity, they recorded from V1 to V4, and the dorsolateral prefrontal cortex. Now, I'd like to draw on a more principled approach in thinking about why feedback processing might be important for the brain and might be important for consciousness. And I'm actually going to draw on integrated information theory. This is a figure from IIT 3.0, as it was termed, published in PLOS Computational Biology in 2014. Now, it's a complicated slide, but the message is pretty clear. You have two systems. One is an integrated system, and it's integrated by virtue of the fact that there are recurrent processes, and the other is a purely feed-forward system. It's important to note that the inputs into these systems are identical. The outputs from the system are also identical. Now, what should strike you immediately is that in order to get the same output from a purely feed-forward system, you need a much more complicated organization. And we would expect that the brain would favor a more energetically and metabolically efficient system, as you see with this integrated, reciprocally connected network. I would also like to note that a surrogate for the capacity for consciousness that's rooted in IIT, it's denoted as 5, was also measured in these systems, and the purely feed-forward system is associated with a 0 phi, so no capacity for consciousness, while this integrated system is associated with a non-0 phi, suggesting that it does have the capacity for consciousness. Now, if these data, or if this theory is not yet compelling, and you should all be very skeptical, I'd like to draw on some empirical evidence from the literature of anesthetic-induced unconsciousness, and in particular, work by Professor Tony Huditz, who's a colleague at the University of Michigan. He published this article in 2005, and this graphical representation comes from an article published in Science by Mike Alkire, Dr. Huditz, and also Giulio Tononi. You're now looking at a rat brain. This is the front of the brain, this is the back of the brain. And you see these arrows in red and green. These arrows are representing a measure of directed functional connectivity. You heard from one of the experts on directed connectivity yesterday, so this should not be completely new. This measure is transfer entropy, and it was in higher frequency activity, so the gamma bandwidth at around 55 hertz. This was in the setting of visual evoked potentials, and you can see in the awake state that there is a balance between this feed-forward processing and this feedback processing. Feed-forward depicted in green, feedback depicted in red. When these animals underwent anesthesia with isoflurane, in the setting of continued visual stimuli, the feed-forward processing was maintained, despite the fact that the animals were unconscious. By contrast, the feedback connectivity, depicted in red, was selectively suppressed. Now, if you turn up the dial of the anesthetic, you're going to suppress all of this connectivity and this activity. But at concentrations consistent with unconsciousness, you see preserved feed-forward connectivity and selective suppression of feedback connectivity. So this is going on in the rodent brain. Who cares? Well, we cared, and we were compelled both by these data and also some of the earlier data, compelled enough to pursue this translationally. So my colleague, Dr. Anshul Lee, a physicist who's in the audience, he and I, with collaborators in South Korea, conducted a series of studies, and I'm going to talk to you about one of them that was kind of the final output in surgical patients, showing that indeed in humans, feedback connectivity is selectively inhibited during anesthesia. Now let me break down this slide for you. The measure of directed connectivity here is normalized symbolic transfer entropy. It's a cousin to transfer entropy. And you see time on the x-axis. These red traces represent feedback connectivity from the front of the brain to the back of the brain. The blue traces represent feed-forward connectivity from the back of the brain to the front of the brain. There's a gray shaded bar you can see for each of these drugs, and that is when the drug was being administered. And thereafter, the patients were anesthetized, as judged by the admittedly imperfect surrogate of loss of response to command. Now, what should be apparent to you is that there is a very consistent selective suppression of this feedback connectivity. In the human brain, in the resting state, and we're not the only ones to show this, there appears to be a dominance of this feedback connectivity. As the drug is being administered, you can see that that feedback connectivity is dropping, and afterwards is essentially neutralized with respect to the feed-forward connectivity. There's no more feedback dominance. Now, one of the reasons why I put this slide up and why I think this is particularly important is because the drug ketamine has really resisted any generalized framework for anesthetic mechanism in the past. It's kind of the problem child of the general anesthetics because it's very, very different neuroscientifically. So at the molecular level, drugs like propofol or sevoflurane to a greater or lesser degree potentiate GABA transmission, GABA being the primary inhibitory neurotransmitter in the brain. Ketamine isn't thought to act like that. It has non-GABAergic targets as its primary therapeutic binding site. If we move to a neuroanatomic target, virtually all sedative hypnotics suppress the thalamus metabolically. Ketamine doesn't. Ketamine maintains oxygen metabolism in the thalamus and actually increases glucose metabolism. Now, if we move to a more systems neuroscience approach, drugs like propofol or sevoflurane promote activation of the ventrolateral preoptic nucleus. This is a sleep-promoting region in the hypothalamus. So when the VILPO is on, we subjectively turn off. And by contrast, they also inhibit arousal-promoting nuclei in the brainstem and in the diencephalon. Well, ketamine does just the opposite. Ketamine suppresses VILPO activity, so it suppresses sleep-promoting activity, and it actually activates wake-promoting activity, despite the fact that it's a general anesthetic. And if you look at neurophysiologic activity, again, very, very different. Propofol and sevoflurane tend to increase alpha oscillations in the prefrontal cortex and frontal cortex. Ketamine doesn't. It suppresses alpha activity, and you heard a little bit about that. Ketamine, along with other psychedelics, suppressing alpha oscillations. Also, ketamine enhances higher frequency activity in the gamma bandwidth, which is normally suppressed by propofol and sevoflurane. So this is a very long-winded way of saying that ketamine is a very, very different kind of drug, and yet coming in to the study with an a priori hypothesis rooted in the neurobiology of consciousness, we were able to make a successful prediction about how ketamine would behave. Now, we're in an era of crisis with respect to rigor and reproducibility in science, and so I just want to note that disruption of frontal parietal connectivity during anesthesia is quite robust and it's quite reproducible. There are many complexities and nuances that I don't have time to discuss. Maybe they'll come up in the question and answer session. But I do want to note that reduced functional connectivity between the frontal area and posterior parietal cortex has been identified by Fmri during propofol, sevoflurane, and ketamine anesthesia. These are independent laboratories. Preferential reduction of frontal to parietal directed or effective connectivity has been identified by EEG, both low density and high density montage, during propofol, sevoflurane, and ketamine anesthesia. And a variety of different techniques of directed and effective connectivity have yielded consistent results. Finally, and importantly, I think, there have been consistent findings in humans, in monkeys, in ferrets, in rodents, and most recently, and I think most surprisingly, Drosophila, which is the fruit fly. Now, you might be wondering, how the fruit fly packs in a prefrontal cortex in that little head of it. Well, it doesn't have a prefrontal cortex, and this gets to the point that it's not necessarily about the prefrontal cortex per se, but about hierarchical brain organization. So there are higher order nuclei in Drosophila brain. They receive feed-forward input from lower order nuclei, and there is feedback at lower frequency, which is consistent with the mammalian brain. A recent study demonstrated that there is a selective suppression of this feedback connectivity in Drosophila brain. So this is really a remarkable finding, and I think very encouraging. Again, many complexities, many nuances, but this is a robust finding. So why should this be happening? Well, I want to share a couple of studies with you. I don't have a definitive answer, but we have to be thinking about what's going on in subcortical regions as well. So you're looking at a study that was combined EEG and fMRI. I was involved in this study. It was with colleagues at the Technical University of Munich, where I spent some time on sabbatical. And what you're looking at on the top panel is again, symbolic transfer entropy, a different kind of representation. So just focusing on the anterior posterior, looking at the side of the brain, the red suggests feedback dominance, and the blue suggests feed-forward dominance. And that, which has no color, is really kind of a neutral activity. So you see in the awake state, much as we found with earlier studies and others have found, that with this particular measure, there appears to be a dominant feedback connectivity. And I want to note, this is in the eyes closed resting state, not during a task. So BS stands for burst suppression. I hope that that's the only BS that you see in my lecture today. And that's a complex state because you have some periods of electrical quiescence and other periods of high frequency, high amplitude activity that can actually be very synchronized and integrated. But you still see a reduction. And at clinically relevant concentrations of sevoflurane, you see this loss of top-down connectivity. I don't have it here, but when the healthy volunteers, in this case, wake up, there's a return to this feedback dominance. So that's the top row, kind of confirming what I showed you before. The bottom row is functional MRI. And now you're looking at a top-down view. So this is the front of the brain, and this is the back of the brain. And you're looking at connectivity with the thalamus, in particular the left thalamus. And when you're seeing this dark, hazy spots or blobs in the awake state, it's because there's high connectivity between the thalamus and the cortex, as we would expect. Now, in a state of deep anesthesia, you can see that there's a breakdown of that connectivity. You see a lot of white patches. But as you come down to more clinically relevant concentrations of sevoflurane, you start to see the return of connectivity to the posterior cortex, but still these islands of white in the frontal area, and at concentrations that are clinically relevant and consistent with unconsciousness, you see really a robust return of a lot of this connectivity, with the exception of the prefrontal cortex. This just points to the fact that there may be some bottom-up processes involving the thalamus that either reduce information flow to the prefrontal cortex, or there could be a lack of coordination between different areas of the cortex, which higher order thalamus is responsible for. I'd like to briefly touch on a recent study that was published from our Center for Consciousness Science. A postdoctoral fellow, Zirui Huang, was the lead author. Dr. Huditz was the senior author, and I had the great privilege of being involved. And I'm only going to touch on this briefly, as Zirui talked about this yesterday in our workshop. But different levels of organization and different levels of connectivity and synchronization were examined. So just focusing on the column of wakefulness, at the voxel level, about 500,000 to 1 million neurons. Autocorrelation in the temporal domain was evaluated. Regional connectivity at the 40 to 60 voxels per area. And then finally, longer range connectivity. And that kind of long range connectivity is important for the global neuronal workspace. And of course, you heard an elegant lecture by Dr. Changeux at the opening plenary session. So look what happens in the state of sedation. So kind of the beginning stages at which you're losing unconsciousness. At the neuronal synchronization level or the regional connectivity level, you actually see an increase So you're not seeing a breakdown, you're seeing an increase in that regional activity with a concomitant breakdown in long-range connectivity. And we can understand this intuitively. If you were having a phone conversation with somebody back home, and then you started to engage in a local conversation with somebody at the conference, you would probably have a communication breakdown with that person that you were calling long distance. Well, something similar seems to be happening in the brain at the initial stages of unconsciousness. You start to get more modular and more tightly synchronized or correlated activity locally with a longer range breakdown. And once you get to surgical levels or pathologic states of unconsciousness, you start to see fragmentation across the board. Now, I want to put this back into the context of what we discussed earlier in terms of this global neuronal workspace framework. This would be the equivalent of, for example, the parietal and frontal cortex becoming more modular, becoming more tightly synchronized within themselves, which would block the kind of long-range communication that might be important for consciousness, leading to a loss of consciousness. And I should note that one of the reasons why this reciprocal activity is thought to be important is because it can amplify and sustain representation even beyond the initial processing at the level of the primary visual cortex. So I now want to move from this neurobiological framework of the network and actually move into some network science. And again, I wanted to acknowledge Dr. Unshul Lee and Junyoung Moon, who was a former postdoc who's now at Johns Hopkins, for their work in this study. So you're looking at another representation of the brain, and in fact, it's the human brain. So forget about the color for a moment and just focus in on these different gross areas of the brain: occipital, parietal, frontal, etc. And each of these little circles in this ring plot represents a particular node in the human brain. These data are coming from human brain imaging, in particular diffusion tensor imaging, which allows somebody to capture structural connectivity. And as was mentioned yesterday, structural connectivity is kind of a basis for some of the other functional connectivity measures that are often used. Now, these black lines connecting the different nodes reflect the degree of connectivity. And what you can notice is that the back of the brain tends to have very rich connections, which in this plot, the precuneus seems to be the most highly connected node. And this is referred to as a kind of hub node. Now, this is a technical term in network science and graph theory, and there's a mathematical formalism around identifying what kind of nodes are hub nodes. But again, we have an intuitive sense of the importance of a hub node because we've all been to hub airports. Maybe you went to a hub airport when you were coming to this conference. And we know that in wintertime, if there's a snowstorm at a hub airport, you get widespread breakdown of air traffic. If you have the similar snowstorm in a peripheral airport, local traffic might be affected, but you're not leading to this network breakdown, so to speak. And something very similar happens in the brain. So Dr. Lee and myself started to explore the notion, and we had some empirical data suggesting it, that anesthetics might create a kind of snowstorm in the hubs of the brain, which would lead to a global breakdown of information transfer. And so what was done in this study is that nodes that had richer hubness were selectively lesioned. So it's like if I were in Michigan, the Detroit airport has this much hubness and the Lansing airport has this much hubness. This airport would get this much snow, this airport would get this much snow. And what we did for both of these studies is we implemented oscillators, a model of alpha oscillation, onto this neuroanatomic scaffold, and then we applied a measure of directed connectivity, directed phase lag index. And that's what you're seeing in the color. So the red is phase lead, that's where the signal's coming from, and the blue or the teal is phase lag, where the signal's going. So what you can see here, just in very coarse terms, is that these highly connected hubs in blue are the targets for activity. These more sparsely connected nodes in the front of the brain are the sources of the signal. And when you selectively lesion these hubs, that whole ring turns to green, which is basically no net directionality. So what I'm trying to express here is that simply by virtue of the network topology, you get this natural behavior of oscillators where you have a frontal to posterior dominant directed connectivity. This essentially recapitulates exactly what I've shown you here empirically. A dominant frontal to parietal connectivity in the waking state that gets neutralized in the anesthetized state, suggesting that hubs might play an important role. So we've talked a little bit about the neural correlates of consciousness and anesthesia, but ultimately what we want to get to is an understanding of the neural causes of consciousness. Now, this is going to be very difficult if you're talking about the content of consciousness or particular qualitative experience, but it might be somewhat tractable if we think about the level of consciousness, being awake versus being anesthetized versus being asleep. And I want to talk about a network involving the prefrontal cortex now, not with other cortical sites, but rather with subcortical sites. So the prefrontal cortex is unique in that it has reciprocal connectivity with a lot of critically important arousal-promoting nuclei in the brainstem and the diencephalon, including the thalamus, the pontine reticular formation, the serotonergic dorsal raphe, the noradrenergic locus coeruleus, the cholinergic system of the basal forebrain, as well as the dopaminergic ventral tegmental area. So there's a reciprocal connectivity. So what we did is we wanted to manipulate anesthetized animals by modulating the prefrontal or the posterior parietal cortex. So they had EEG, they had microdialysis catheters either in the prefrontal cortex or in the posterior parietal cortex in two different areas. They were anesthetized, and then we reverse dialyzed either carbachol, which is a mixed cholinergic agonist, it's ramping up the acetylcholine receptors, or noradrenaline, and then we observed behavior. Now, I want to note that I'm going to show a video of some animal experimentation. I'm just warning you because I know some people are not used to this kind of video in this kind of conference, so I'm going to be showing you in a moment. But this is the experimental setup. The rodent is in a sealed chamber where it receives oxygen and sevoflurane. It has EEG signals that are going to this computer, and it also has a microdialysis catheter where we can deliver drugs and also sample acetylcholine. And what we found, first of all, when looking at the prefrontal cortex, is that cholinergic stimulation in anesthetized rats could restore wakefulness. And I'm going to show you that video now comparing prefrontal and posterior parietal cholinergic stimulation. Surprisingly, the norepinephrine or the noradrenergic stimulation, although it changed the EEG, didn't seem to change behavior. So this is the video, and you're going to see the animals breathing in, well, you're not going to see them breathing in the molecules, but they are anesthetized, they're breathing in sevoflurane. You can see the EEG tracing up here. This is the EEG signal. You see that little catheter. And this animal has had carbachol in the prefrontal cortex. This animal has had carbachol in the posterior parietal cortex. Now, pretty soon, you're going to see the EEG getting more narrow, getting higher frequency, lower amplitude, suggestive of a more wakeful state. So just observe that for a moment. You see a little epileptiform activity that's happening here with this posterior parietal cortex stimulation. And now note the respiration increase. And this is interesting because you see this EEG activation. The animal still appears to be unconscious, but you can see there's some top-down signaling going on. So now the brainstem is being affected by that prefrontal cholinergic stimulation. The respiration is increasing. So let me just jump ahead. And you can see this animal is now trying to turn over. So how do we know an animal's unconscious, a rodent? We look for the righting reflex. Animals don't like to be on their back. They always try to get on all fours. And when they lose that righting reflex, we consider them to be unconscious. So the animal is now trying to right itself, and in fact now has the recovery of the righting reflex. Now keep in mind that this animal is still breathing in clinically relevant concentrations of a general anesthetic and appears to be quite active despite the fact that the concentration of anesthetic hasn't changed. So to conclude, reciprocal activity across frontal and parietal cortices appears to be important for the contents of consciousness. This has been discussed in the context of access consciousness, what we can access in order to get something into working memory or verbally report, but there's also evidence of the prefrontal cortex being important for phenomenal consciousness. This reciprocal activity is selectively suppressed by diverse general anesthetics. Again, there are many nuances and complexities, but it's been a fairly robust finding. And activation of the prefrontal cortex can reverse anesthesia and thus might regulate level of consciousness. And I want to note, there are different questions on the table. I mean, with phenomenal consciousness, we want to know what is the neurobiological substrate of having even one pixel of experience, just a completely dark room and one photon that we can register subjectively. Access consciousness is wider and it's richer, but the prefrontal cortex is pretty interesting because it seems to be a hub that can be involved in phenomenal consciousness, in access consciousness, perhaps regulate level of consciousness, and also can drive attentional processes. So it's a hub that can control a lot of different dimensions for being conscious in the way that we're conscious right now. I'd like to thank my wonderful colleagues at the University of Michigan, the Center for Consciousness Science. I've already talked about some of these individuals. I didn't mention Dinesh Pal and John Dean, who had done that work in reversing the anesthetized state through manipulation of the prefrontal cortex. I'd like to give a shout out to our graduate students, John Dean and Michael Brito, who are currently in my lab. I like to surround myself by hair. It just makes me feel more secure. But, John has done some wonderful work. You heard about some of his work from GMO yesterday. He's had some recent articles accepted. Michael Brito just got an NSF grant for his graduate school career to study consciousness. Nick just got into a pharmacology program at the University of Michigan to get his PhD. All of these individuals are really passionate about consciousness. Makes me really excited to see this next generation coming up. I want to acknowledge my funding sources, and I also want to note for the graduate students and the postdocs and the junior faculty that you can, in fact, have an NIH-funded career as a consciousness science. Now, I'm not putting hard problem in the title of my grants, so you've got to be a little bit creative, but, for example, this grant with Rick Harris, who's here in the audience, we're studying network-level mechanisms of ketamine and nitrous oxide. We've been able to incorporate psychedelic studies while still delivering on the NIH promise. And with that, I will thank you for your kind attention. It's a pleasure to be here, and I look forward to addressing any questions you might have in the subsequent panel. Thank you.
As George said, I forgot to mention at the beginning, we'll have, as with the other plenary sessions, we won't take questions in between talks, but we will have an extensive panel discussion at the end. So save your questions, and please remember George's talk so that you can ask questions about that, even though it would have been a little bit later by the time we get to the panel discussion. Now, it's a great pleasure to introduce our second speaker, Yan Xu from the University of Pittsburgh, and he is going to come and talk to us about non-deterministic information access as a basis for consciousness. And there might be just 30 seconds while we have to swap the machine. Would you? So I'm not sure what we have to do here. It's just the HDMI. Oh, it's already plugged in. Okay. Great, there you go. Great, thanks.
All right, so it's really a great pleasure to be here, and I'd like to thank the meeting organizer for this invitation. And so today I'm going to tell you a little bit about what we have been working on in my lab at the University of Pittsburgh. And many of us come to this conference with the hope that we will be able to shed some light on the problem of consciousness. And we also come here to prepare to accept our difference in the philosophical views of consciousness and neuroscience view of consciousness. But in any case, I'm particularly putting two different colors there, and some of you will be able to tell the difference. And so phenomenological consciousness is actually our inner experience of it. And yesterday afternoon and this morning, there's beautiful talks about the AI, the artificial intelligence, and the virtual reality. And so I don't know, with this epigenome change, will future generation much smarter than us by being able to perceive on the phenomenological consciousness of other dimensions? I don't know about that. But let's look this afternoon, focus on the neuroscience aspect of it. And as we know that we talk about wakefulness, self-awareness, and as Dr. Mashour mentioned, that there's access consciousness, and there's also the content of it, the learning and the memory aspect of it. So today I'm going to tell you a little bit of the study that we have been working on this domain for many years. And as we know that the neurons are connected, and so we pay a lot of our attention on the way that how the neurons are connected. And my lab, in collaboration with Peyton's lab, have been working on this problem of looking at the high-resolution structures of these proteins that control the propagation of information, and also the receptors that control the signal synaptic communication. And we also like to throw a lot of anesthetic molecules onto these receptors and to find out what happened to these receptors. But today, I'm going to focus on a broader picture, and we like to understand what is the state of mind under general anesthesia. And in fact, we actually don't know what exactly state that a person under general anesthesia. And empirically, we made a lot of assumptions, and we assume that the person is unconscious based on our empirical definition of consciousness. And we also know that the patient will be amnesia. They have no recollection or memory of what's going on under general anesthesia. And we also assumed that the brain under general anesthesia is incompatible with information process, or at least the information process is being dampened. But rigorous experimental tests of these assumptions are actually not are still needed. And so before we really can address this issue of neural correlates of consciousness, and also I listed here as neural correlates of unconsciousness, there are some very fundamental questions that we have to answer. So as Dr. Mashour alluded to just in previous talk, that there's a conscious content. And so when you have general anesthesia, and what is that content we are talking about? And we also ask whether anesthetized brain can continue receive information. And based on the circuitry, we already know that in the there's a feed-forward process, and we probably all agree that the sensory input are still reaching different part of the brain. So the brain is still receiving information. And can they process and consolidate the information? This becomes murky already. And if they can perceive external stimulation and make a record of it, and that will be something that's against our common belief. So our intuition tells us that if animal or human under general anesthesia, in the surgical level of anesthesia, they probably will not be able to process the information and until they wake up. And so if you have experimental data that shows you that by letting the animal, in this case, to learn some tricks under general anesthesia, and you forever altered their ability in the future to learn that trick, then you start to think maybe there's something happened under general anesthesia. So this is what I'm going to tell you. And what I'm going to show you, again, also 2 short videos of animal behavior. And so I understand that in this conference, as Dr. Mashour mentioned, that people probably will not feel comfortable seeing the video of animals. But this is just naturally behaving animals. And so what we're going to focus on is the olfactory information processing in rodents. And as we know that the rodents use smell as their conscious behavior. And if a mouse found a way to an open jar of peanut butter that your kids left on the countertop, and you know that mouse is conscious. And so we focus on the behavior aspect of it, and to see what conscious animal will do when they deal with some information that related to olfactory learning. Then we also have a way of looking into their brains to look imprints of their learning. And so this is one you gave a rodent, a rat in this case, some odor which is novel. So this odor that this animal has never experienced before. Then this animal will go over to spend a lot of time investigating the odor and try to understand the odor. And so I can let it run. It will run a long time, and the animal will stay there. They're going to walk away. They're going to come back to the same odor just to learn it. And if there's no incentive associated with this odor, then the next day, this odor already become familiar to the animal. So next day, if you gave the animal this exact same odor, and as shown in this video, it gave this animal the same odor, the same way, and they detected the odor in the cage. This heading, the rearing is the detector of odor. Then the animal will walk away, and they are not going to go back, approach the odor. And so this is very distinct behavior endpoints of whether the odor is familiar to the animal or the odor is unfamiliar, novel to the animal. So of course, in doing the experiment, it's much more complicated than just putting an odor into the cage. So this is the cage that we actually designed. It's a black box, it's an open field. The animal has a tendency to running around. I think the mouse doesn't really show over there. Try to. So the animal has a tendency to move around the edge of the cage, but inside here you will now see is a hidden container that has different odor that coming out from it. So if it's a novel odor, the animal will have the courage to run into an open field to smell it. And if it's familiar odor, they will not spend time to the center of the cage. And all this behavior is recorded automatically, and then we can analyze afterwards. So in this case, we will be able to compare 2 things. One is the behavior that the animal tells us whether they learned and are familiar with the odor. And then we can also doing this study of using immediate early genes and also learning associated immediate early gene to see what's going on with the brain when you have the animal learn different tricks, different odors. And so in olfactory system, and there's an advantage and disadvantage to use olfactory system. Olfactory system bypass thalamic cortex pathway. So that in that sense, it's different from the visual cortex. But the advantage of it is that you can simplify things a lot. And so-called feed forward in the olfactory system is actually feeding back. So this will be the primary sensory neurons. It's coming to the peripheral cortex and going to the orbitofrontal cortex. So feed forward is actually going this. So I think I have slides later on. So the experiment design is actually quite simple. So you will have, there's many groups that you can think of, but the core group are such a situation. So you will have training, let animal learn something, then you start to test them. And you can do it, do the experiment with the animal fully awake, and you can also do the experiment with the animal in the surgical plan of anesthesia. And during this anesthesia or awake state, you gave them different scent, different odor. And then, you can also, as a control, you could give them nothing, and you look at the behavior of it. So this slide summarizes the behavior response. So when you gave them, the axis reads, so this is during the training phase, this is during the testing phase. So if you train and test with nothing, the animal, will, of course, has no interest in the center of the open field because there's nothing encourage them to go to the center. So their time spending in the center is very, very low. But now if you give, in the training, you don't give them anything. Then you give them a novel odor. And you can see that they start to come into the center of the cage, start to investigate the odor. But now if you have this animal already learn that odor, and you do it again the next day, they spend almost the same amount of time as if there's no odor there. And so that means familiar odor means nothing to this animal. And this purple bar only to show that the anesthetics, when the animal exposed to anesthetics, doesn't change their behavior if you gave them the odor the first time. That means the anesthetics does not cause brain damage. In this particular experiment, we use ketamine, the old child of anesthetics. And so they did not change anything. You train with nothing, but anesthetize them. Then you next day gave them a novel odor. They come out investigated. But look at this one. So this one is when you pair the odor with anesthesia. So the animal is fully anesthetized. Then you give them odor. And when they wake up, just like a human experience, they don't know that odor they have already experienced before. They have no recollection of memory. So this is the novel odor, this is anesthesia paired odor, and there's no explicit memory of that odor. But how about, so this is what I was talking about. So this is feed-forward process. So the information is flowing this way, coming from the olfactory bulb and going into the anterior peripheral cortex. As the secondary, this is where the information is consolidated, then it's projected into the optofrontal cortex where the information is being processed and it's being perceived. And so now when we look at this situation with the, in this particular case is CFOS. CFOS is a good indication of the neuronal activity. So again, with training with air and then you test it with air, the CFOS level, because there's nothing more than the background of the environment that the animal is in. And their CFOS level is at around, this is 200 cells per millimeter square. And if you give the animal a novel odor, and you can see that the CFOS level immediately increases. This is in the peripheral, anterior peripheral cortex. This is in the optofrontal cortex. And in both situations, the CFOS level is greatly increased. And now if you look at familiar odor, and the CFOS is no longer turned on. And this is information that's actually very useful. That tells us that with the same odor, the animal will use the same set of neurons for that particular odor. If you give a different odor, the bar will go higher. But if you give the exact same odor as a familiar odor, it's come down. So that means there's no neuron that's being involved in the CFOS activation. But look at this purple bar. Again, the anesthetic didn't really impair the ability for the CFOS to be turned on. But remember that this gray bar was as high as this one, but in the brain now, the brain says that I already know this odor is already processed before. So the CFOS is no longer turned on, even though the first exposure of this odor is on the surgical level of general anesthesia. So Then we will say why the animal cannot recall the information that's already stored in their optofrontal cortex. So maybe we should change our design. And in the human memory study, there's this method in human studies called the familiarity test. That is to compare something already studied with something unstudied, so never studied before, so that the human behavior will using something in the cognition memory of the familiarity part of it to tell you whether something is familiar or not. So over here, we will have one odor. We call the odor A, which is the odor we use to train the animal. Then we compete that information with a brand new odor, we call the odor B. And in this case, the two odors are a banana odor and citric odor. And we select them because the animal have no preference of either one of them. And we counterbalance it, so we change whether banana is odor A or citrus is odor A. On different animals, just randomly switch them. And then you videotape the animal and look at how much time they spend on each one of them. And to make a contrast, these two beads are the beads from the cage of the animal. So it's a background odor, and they have been experienced. And so that will increase your power in the analysis. So this is a flow chart, and I color coded in such a way so that you can follow in the subsequent slides. The blue is what we call the learning. So that's the initial learning, that you have animal exposed to some novel odor. And some of animals were sacrificed, and we look at their immediate early gene. In this case, it's the learning associate NPAS4. So it's a neuronal pass domain 4 immediate early gene. So that gene will turn on if there's a novel information to be learned. It modifies the inhibitory neuron on the excitatory spine, so the excitatory axonal spine. So it will modify the spine. And so that's, that gene turned on, you know the animal is learning some new things. And so some of the animal will be sacrificed, and we're going to do NPAS4 staining. And the other animals will continue on to look at their cognition test of the comparison, the familiar test, which I call it the forced choice test. So you you force the animal to choose one of the other. Then during this phase, they are all awake, right? And they also will learn this odor. Then you sacrifice some to do the immunostaining and leave others to go on to do a second phase of evaluation. So this is called a relearning part. The red part is relearning, and the blue part is initial learning. And so let's look at the optofrontal cortex of the NPAS4 during the learning and relearning. And initially, so this is the control. When you have control, this is without primary antibody, so there's really nothing, right? And this is the background level of the NPAS4 that is expressed in the animals. In their daily life, they learn something and they turn on their immediate early genes. So these are the background level of the NPAS4 that for a normal behaving rodent. And so we take this line. This is a 2 standard deviation outside the mean of the activity of the counting of the NPAS4 positive cells. And this is just view it as flow cytometry, but it's basically the same technology. But it's done in the immunohistochemistry method. But now, if we do, I'm sorry about the color. So this is the blue. That means it's a learning phase. As you can see that when the animal is awake, you give them a novel odor. Their NPAS4 is elevated significantly. But look at this one. This is under propofol gen anesthesia. So it's at the surgical level of a propofol gen anesthesia. And we know that propofol is GABAergic, many GABAergic anesthetics. So under propofol gen anesthesia, you would expect that because of the potentiation of an inhibitory neuron, you don't have information processed there. But if you look at the learning behavior, indeed, they still turn on the learning-associated immediate early gene. But now, when you do the relearning process, look how fast, because during the relearning, remember, animal is awake. In both cases, in both groups, the animal is awake during relearning. But they no longer, in the awake group, they no longer turn on the MPAS4 because they learned it before consciously. But this group, they learned it under general anesthesia, so they are unconscious while the first learning occurred. But they, again, will not turn on their learning-associated gene, as if. And this is, again, as I said, no new neurons are involved in learning that particular odor. And so this gave us a question. Apparently, brain is modified. And our question is, if the neurons that encodes the initial learning has been occupied, even under general anesthesia, but they have no recollection of that odor, and will they be able to learn that odor again, right? So that's a fair question, and they ask the exact question about what is the content under general anesthesia, so this is where I'm using bar graph to summarize what's going on. So this is a control group. So this is the green, the blue means that it's in the initial phase, the first learning. And the control group, they never experienced anything. So it gave novel odor. A and B are both novel. They spent about 50% time on these two odors. And these are the beads inside the cage, so they show no interest in those beads. So this is the control. You know that the animal has no preference for either citric or banana, and it is counterbalanced. So they spend about 50% of the time on either of the odor. But now, if the animal has the previous experience with the odor, so this odor A always is something that they already learned before. And you can see that they spend much more time on the new competing novel odors, odor B. And so they spend much more time on odor B than odor A. And again, with anesthetized brain, even though their MPAS4 level come down to the same level as awake, they, again, have no recollection. Even you have a brand new competing odor there to compete with, they still report that there's no familiarity of the odor A. So that tells you that, indeed, the animal cannot remember it. So now we move into the relearning part of it. So if, remember, the second learning time, the animals are awake, so they don't have anesthesia during the relearning phase. And the control, you will have, they still like to, because those beads have odor. And this, the same with the odor, they are learned twice, so they still have equal preference. But pay attention to this particular one. So this one, they actually exposed twice. The first time is under anesthesia. And remember, the second time when they learn, they cannot modify their MPAS4. Their learning-associated gene is no longer turned on. And they also learned odor B during the learning, right? During the relearning, they also learned odor B. And now, they do not spend time on odor B, but they have a lot of difficulty recalling odor A. So that tells you that when the odor was experienced, learned the first time under general anesthesia, the loci of this imprint of this odor will not be released. So the animal will have hard time to learn that same odor even they are awake. So this morning when I heard the MIT student start to learn things while they are half asleep, and I don't know what will happen to their brain afterwards. And so this is the difference between the two groups. And as you can see, there's significant impaired. And so now let's look at odor mixture. So this is one area that we are struggling a lot, because when you have a pure odor, there's really no association with the odor. But if you have a mixture of two odors, now, let's say you have banana and citric, and you let animal to learn this mixture of the odor. And will they be able to find out this odor actually are learned before? And so in reality, it's this. During the initial learning, again, if you gave them odor mixture that they learned before, and they already know the content of one of the component in the odor. So this is novel. So you have citric and banana mixed, but then you use citric as independent odor. And they say that citric is the same as citric and banana. So they spend the same amount of time over there. The anesthetized animal is the same way, but they don't have recollection. But interesting part of this is that during the relearning, so this is during relearning. During the relearning, the animal actually relearned the association of the A and B, the two odors. And they also learned the odor B by itself. Then during the relearning behavior test, you found out that they actually have no interest in the mixture anymore, but they show a lot of interest in the odor A itself, as if they have never experienced the odor B itself, as if they have never experienced it. And if you look at the difference of the two, this time it's reversed. Since, in interest of time, I will just this is a take-home message from this experimental data. That basically tells you that there's a rich neural content, even under unconscious state. And we also know that the unconscious learning actually prevents the future learning. And the anesthesia blocks only the information access. And since I'm running out of time, I will skip through a few slides that are actually covered by Dr. Mashour's talk. So this is the center where it's a feed forward and feedback. And we also believe that there's this inhibitory from the VRPO that inhibits all these arousal centers that is believed associated with anesthesia and is believed associated with sleep. But what I'm going to present to you is that this deterministic view of the projection into, so this is the arousal pathway of excitatory projection to the cortex, and also it projects the same neuronal transmitter, but it's inhibitory to the sleep center. And during some kind of unknown process that we believe that the inhibitory pathway is being turned on in the sleep, and presumably also in the anesthesia state, so that they are going to go into an anesthetized state and the loss of consciousness. But since in the interest of time, and I only have according to this clock, I have 6 minutes left. So I'm going to leave you with this thought, how we are going to understand the brain under general anesthesia and the correlates of unconsciousness, as particularly on the UN part of it. We have to actually come out of this local field. So this is bolts and nuts that we pay a lot of attention to it. But you probably even do not want to focus on this scale, and even this scale. And we have to come out to really look at this scale. And so this is what we have been working on in my group on the theoretical aspect of it. So we look at the information that feeds into this one, how it's being percolated through the brain in through this network. And I want to draw your attention to these two figures. They look the same, but in our theory, they can represent one input layer in the brain, and you can have output layer, which can be a cortex, can be other regions. But you can also have this model to tell you what is the past and what is the future. So the brain should have ability to do that. I'm not saying that they can predict the future, but the theory will tell you. So this is for those who are interested in this one, this is a paper that we published 2 years ago on the theoretical account of how consciousness is being percolated through the brain. And there's nothing, fixed connection in the brain is not required. for us to really achieve unconsciousness. And I will not bore you with the equations, but the equation actually is quite simple. And so we test the theory based on the human data. So this is the EEG that has been published in the literature. And so we were able to look at these EEG characteristics: frequency shift, synchronization of different parts of the brain, and burst suppression during deep anesthesia, and anterioration. And if you look at our model that predicted, and these are the information coherence when you look at the input signal versus the cortical signal. And this is, you can also look at from cortical nodes with cortical nodes. And the coherence are exactly the same, and this is anterioration. So I will skip through this one. I just want to leave you with one visual experience of what's going on in your mind when this theory actually works. So this is actually calculated from our model based on the entropic change of the information in the picture. So I'm going to, so forget about this one. Those who are interested can read the paper. So this one is actually a picture that's inputted into a node. Then we look at the cortical node, and nobody can tell what's going on because this one, the percolation probability in the network is 0. And so you increase it to about 3.3. So 30% of the information can pass along. And can anyone tell what picture is this one? So people already get, so this is only 0.02 difference. And so this is the way that your brain is actually processing information. So you just need to have a very small change in the ability for the neuron communicate, the probability of neuron communicate. You start to have perception of the information of the outside world. So I'll leave you with this quote from Einstein. And thank you very much. I'm sorry I ran a little over time, but these are the people who did the work. And I'm pretty sure many of you will experience some mind switching because your mathematical mind and your literature mind is doing things. And you are going to suddenly realize your brain is actually working. Thank you very much.
Okay, so it's a pleasure to introduce the final speaker in this session, who is Dr. Marine Vernet, who is currently working at the NIMH in Bethesda and the laboratory of Leslie Ungerleider. And she's going to talk about disentangling models of visual awareness. I think we probably, oh, it's on this, is it? Okay. Do you need, is it lightning? This is the one that's, so you can just take that. Okay. Thank you. Sure. That's it. Let's see if this is going to go. So yeah, don't ignore that clock. This is fine. So I'm going to wait 5 minutes. All right. You have to use that. Let's move it over here. Okay, use the microphone. Okay. Thank you.
Hi everyone, my name is Marine Vernet, and it's a real pleasure for me to be here. I would like to thank the organizers for inviting me, and in particular George Mashour. The Science of Consciousness is a conference I wanted to attend since a very long time, and this is the first year I'm finally able to do so. So this is for me a real pleasure to be among you and to learn so much from your own, who are all passionate about consciousness. I would like to start by showing you this sculpture from Roy Lichtenstein that you can admire at the National Mall Sculpture Garden. in Washington, D.C., where I am currently living. When you are in front of this sculpture, you see a cute little house. But if you turn around, you quickly realize that it has nothing to do with a house. First, it is not convex like a real house, but it is concave. And then this middle edge that you thought was pointing towards you, it is in fact pointing backwards. It is all quirky, and it has not a single right angle. So probably you were misled by the fact that this edge was longer than the side edge, and also by your a priori knowledge about house. But the interesting thing is that if you come back to your original position in front of the house, you can't help to see again an almost normal house. Even if you are now aware of small weirdness, for example, how the house lay on the ground. And this is to me something that is fascinating, that you can have both at the same time, a subjective feeling which is very strong of seeing a house, and this objective knowledge that you also have, that you are in front of two strangely shaped metal or steel plates. And this dissociation between the subjective, the strong feeling, and the objective, the knowledge, is what has been fascinating me the last few years. So there are many definitions of consciousness and awareness across philosophy and science. In my studies, I'm referring mostly to the subjective feeling of seeing, sensing, thinking, etc. And I will be mostly interested in visual awareness, or the awareness of visual information. And through my talk, I will make the dissociation between this subjective feeling that I call awareness and the objective perception, which is how right we are about what we see, how much what we see corresponds to what it is outside in the world, at least as we can measure it with other instruments. I'm going to present you two studies today. Both are done with young healthy participants, around 24 subjects for each study. The first one will be a behavioral study where we will try to dissociate the effects of attention on awareness and objective perception. Attention is a selection mechanism, so we all know more or less what it is. If I ask you to pay attention to this slide, you will probably perceive it better, process the words that are on this slide, and maybe become conscious either of how they look or of their meaning. You will perceive them better and become maybe conscious, but we are going to study in this study the effects of attention simultaneously on these two aspects of perception. In the second experiment, we are going to use functional magnetic resonance imaging to dissociate the brain networks of awareness, objective perception, and also confidence, which is how sure we are about what we see. And by isolating these three different networks, we are going to select the one from awareness and use that to disentangle different models of visual consciousness that has been proposed in cognitive neuroscience. I'm going to start with kind of simplistic models of how we often understand visual consciousness. There is some visual information in the outside world that enters our brain through our eyes. And then this information travels in different areas of the brain, make its way to more and more complex processing areas. And at some point, the information is selected by attention so that it can be perceived better and enter awareness. And indeed, previous studies have shown that attention can improve objective performance and also influence awareness. And in this particular study, we are going to examine how much attention impacts both, if there is any relationship between the impact of objective performance and awareness. For this, we used a visual task, an attentional task. For those who are familiar with the literature, it's called a Posner task, by the name of the person who introduced such kind of task, where we have an attentional cue, in this case an arrow, that indicates to the participants where they should be paying attention to. So the arrow can be pointing towards the left or towards the right, indicating to the participants that they should pay attention to the left or to the right, in that case, to the right. After a short delay, a target appears very briefly. The target can be in the attended location, meaning that it appears where the participant was paying attention to, where the arrow was pointing towards a little before. And this is the case in 80% of the case. So most often. A little less often, in 20% of the cases, the targets appear at the unattended location. So in this case, the participant is paying attention to the right and the target is appearing on the left. The target is briefly flashed, and in addition, it is covered with noise, so that it is really difficult to see. And we made sure it was difficult for every participant to see by adjusting the level of noise to what we call the perceptual threshold, meaning that if the subject has to guess, they will see the target half of the time and don't really see it half of the time. The target was made of lines that could be tilted to the right or to the left. The task of the participants was to answer how were the line, if they were tilted to the right or to the left, but they had also a third possibility, the possibility to answer, I don't know. And the fact that they give one single answer among three possible ones allow us to estimate measure of both objective performance and visual awareness. And I'm now going to explain you how. So for this, we used models from the signal detection theory. So to give you an intuition of how the signal detection theory works, let's say we have in the brain two kinds of signal, each representing the two targets, lines tilted to the right and line tilted to the left. These signals can be anything. Let's say for the sake of simplicity, let's imagine that it is the activity of a single neuron. So on the y-axis, we see the firing rate of one neuron. If the neuron fires a lot, like here, it's most likely the right-tilted target. And if the neuron doesn't fire a lot, it's most likely the left-tilted target. And if the neurons fire somewhere in between, we can't really decide. It can be both. If the two brain activities encoding the two targets are largely overlapping, are close to each other, it's difficult to choose in between the two targets. We will say that the objective performance is quite low. On the contrary, if the two distributions are further apart, it's easy to distinguish between the two signals. We will say that the objective performance are quite high. So the distance between these two distributions represents the objective performance, or truly we are able to see. But the question to our participants was not only to say if the lines were tilted to the right or to the left, they also had the possibility to answer, I don't know. And for the same level of objective performance, the amount of time they are using this I don't know option will be consistent with the awareness. For example, if they choose to answer very few times I don't know, we will say that they are more aware than if they choose to answer a lot I don't know. So the beauty of the signal detection theory is that we can measure independently the objective performance from the awareness. So back to our task, we have some targets that are attended, targets that are unattended, and we use for both kind of targets the signal detection theory to measure objective performance and awareness. And we found that for those particular type of targets, attention had no effect on objective performance, meaning that it doesn't matter if the target was attended or unattended, we don't really see better. So this is a bit unusual. Usually, attention increases performance. This is related to the fact that we use those specific targets embedded in noise. We can discuss that later, but I would like to point towards the most interesting result of this study, is that despite the absence of attention on the objective performance, attention increased visual awareness. So when the target was attended, participants used less the I don't know option than when it was unattended. In other words, with attention, participants believed they saw better, even if they truly did not. They kept the same level of performance. So this dissociation between the effect of attention on objective and subjective performance called for a reevaluation of the simplistic model of visual awareness. Probably the visual information that enter consciousness is not just selected information, objective information that has been selected by attention, but there are many more components that are involved in this awareness buildup. And one of them, the one we reveal by this study, is that probably we use the knowledge of where our own attention is. So in our study, when the target appeared where we were paying attention to, we believed we see it better. So we use this knowledge of our own attention state to build our consciousness content. So I'm now going to switch gears and present you the second study, which is a functional magnetic resonance imaging experiment, where we again dissociate awareness and objective perception, but this time we aim to dissociate the neural networks of these two components and also of another closely related function, which is confidence. Confidence in what you just saw. And we used this dissociation to isolate the networks of awareness and disentangle models of visual consciousness that have been proposed in the literature. Before I'm introducing you the models, I would like to show you some important aspects of visual perception. So for those of you who are not yet familiar with this image, I don't know if it's many of you, you might be seeing just some random patches of black and white in this screen. And I'm going to change the way you see this image, and most likely forever, by telling you that there is a dog, a Dalmatian, in this image. So here is the head of the dog, here's the body, the legs. So I guess now everybody sees it, and if not, you can ask your neighbor who probably knows where the dog is. The point is, now that you see the dog, you can't go back to seeing only black and white dots. So, and even if you meet this image in 5 years from now, most likely you will see the dog again. In that case, perception acts like an all or nothing phenomenon. Either you see the dog or you don't see. You can't really go in an in-between situation. But there are other cases where perception can be more gradual. For example, if you present visual stimuli with a short or longer duration, and you give to participants a perceptual awareness scale, where they have the possibility to answer, I didn't see anything, I had a brief glimpse of what it was, I had an almost clear percept of what it was, I have a clear percept of what it was. Participants indeed used all these levels. So for very shortly presented stimuli, they will say that they didn't see anything most of the time. For longer durations, they will have a clear percept. But for intermediate durations, they might use this intermediate rating as well. So in that case, perception can be gradual. So we have these two kind of opposite aspects of visual perception, and one way to reconcile it is to, again, call the attention. So attention, as we saw earlier, can enhance conscious perception. But when a percept is identified, when it is recognized, it can further grab the attention. And we have a reinforcement loop that might be important to stabilize the percept. So you don't have to, again, try to recognize something that has been once identified. So you keep the percept, you stabilize it. So this aspect might tell us that we have two different things going on, one which is a gradual buildup of awareness, and one which is related to this identification mechanism, stabilization mechanism. And both can coexist at the same time. One will be more related to the subjective feeling of perceiving, and one will be more related to this objective identification mechanism. So in this study, what we are going to do is we are going to look for signature of these different patterns, and we will look for we will compare the patterns of awareness to the one predicted by models that are available in the literature. So I'm going to present you now 3 models. I chose those models because they made clear prediction on where we will find awareness-related activity, and they are all based on philosophical approach of consciousness. So the first one is based on Barr's theater of consciousness. So in that case, the theater is a metaphor for awareness, where we have a stage and the attention shines a bright spot on the stage where actors that are cognitive processes come and go, and the audience are the rest of the brain, the other unconscious processes that benefit from the information that is played on the theater. And the neuroscientific proposal that implements the theater of consciousness from Barr's as being proposed by Dehaene and Changeux. It's called the global workspace. So we already talked a little bit about that in the session previously, but for example, for visual information, we can say that it enters the brain and goes in early visual areas and then make its way up to the hierarchy of the ventral stream, the dorsal stream, but it can stay unconscious. It can be processed quite far in this hierarchy of brain, but stay unconscious. But it's only when this information reaches the frontoparietal network that there is a reverberation of information between these different areas and a maintenance on the information long enough so that it can become conscious and access to the rest of the brain. However, to researchers following net block ideas, such frontoparietal activation might rather reflect access consciousness, which is information that is accessed by the executive system so that one can act on it or report it. But there is another form of consciousness, which is phenomenal consciousness, which refers to the subjective feeling that we are all interested about, and this might have a different neural substrate. So Lamme has proposed along this line that indeed when information reaches the frontoparietal network, we are most likely having the access consciousness type of experience. What matters for phenomenal consciousness is that we have recurrent processing. So let's say that information just enters into the brain and then goes from V1 to V2 to V4, etc., up to the hierarchy, but just in a feed-forward way, it will stay unconscious. However, if we start to have reverberation between these different levels, that's when we will start to build up consciousness, in that case, phenomenal consciousness. The last model I would like to introduce as being proposed by Gradual. The proposal is that consciousness is a model of our own attention state. We constantly track to what and where we pay attention to. And this model of attention that we maintain constantly will be awareness. And when we track what and where others pay attention to, we attribute consciousness and intentionality to them. And it has been proposed that such model of attention, which is awareness, will be implemented close to the temporoparietal junction or the superior temporal sulcus, because those areas are important for both perception and also social cognition. So those are our different predictions. We will look for awareness-related activity and check if we will find such activity in the frontoparietal network, or if this activity will be limited to the ventral stream, or if we will find such activity in the temporoparietal junction, superior temporal sulcus. So for this, as I said, we used a functional magnetic resonance imaging experiment. And again, we had a visual task where we showed participants images of face or avocados. Again, the images were very briefly shown and covered with noise. They were very difficult to see. We were at perceptual threshold. After a short delay, the participant could give the answer. The answer could be, I didn't see anything, I saw something, but I'm really not able to identify what it was. I saw a face on avocado, but I'm not really sure. Or I saw a face on avocado, but I'm more sure. One more, the participants give us one response among several possibilities, 6 of them, and this will allow us to recover both the networks of objective performance, that is to say identification, of visual awareness, and of certainty. So again, for the same type of stimuli, very difficult to perceive, we have different subjective answers possible. Nothing, something, but I don't know what. In this case, an avocado, but I'm not really sure, or an avocado, but I'm more sure. Any brain areas that closely follow these ratings will be associated to awareness, this awareness build-up that match the subjective ratings. On the other hand, any brain areas that have the more dichotomic type of activity with more activation when the percept is identified than when it is not identified will be related to this identification mechanism, stabilization mechanism. And finally, we could say that we can also recover from this data a third mechanism, which is the one of certainty. Indeed, our participants, of course, were more sure when they answer, I saw a face on avocado, I am more sure. But after the experiment, they will often tell us, I was sure I didn't see anything when I pressed the button, nothing. So those two extreme ratings were associated with a more certainty about one's own percept than the two intermediate ones. Those are our results. So these are the activity associated to awareness, identification, and certainty. And I'm going to walk through each of those one by one. So first, we found activity related to identification in parietal areas and also a little bit in prefrontal areas. And in both hemispheres, but mostly in the right hemisphere. This is compatible with the fact that it is probably an attention-based mechanism, because attention is known to be important in frontoparietal areas with the right hemisphere dominance. We found also some activation in early visual areas. So to orient you, this is a ventral view of the brain. So like if we were taking a picture from below, this is the back of the head, and this is more anterior. So this is what we call the ventral stream. Early visual areas are in the back, and more complexly, or areas processing more complex percepts are more anterior. So we see some dichotomic activation in the early visual cortex, which is consistent with the fact that frontoparietal network exerts some feedback over early visual areas. Concerning the network of awareness, we found it widely distributed in both hemispheres, right and left frontoparietal cortices, and also along the ventral stream. So this wide distribution of awareness-related activity is most compatible among the 3 models I presented to you at the beginning with the global workspace hypothesis. Also, there are some slight differences. We didn't find much activation in the dorsolateral prefrontal cortex, which is an important node of the global workspace. And also, the global workspace was hypothesized to work in a more dichotomous fashion. Either the information reaches the frontoparietal network, and then there is an ignition and a reverberation on the information, and it can become conscious, or the information doesn't reach the frontoparietal network, and it stays unconscious. So in our case, we showed that it can be more gradual than it was initially hypothesized. And finally, this last network is the one of certainty. We found such U-shaped activity in the left, close to the left superior temporoparietal junction, and right superior and also middle temporal sulcus. So those areas would not be associated directly to the subjective feeling of perceiving, to awareness, but more to the certainty, which is a metacognitive process of evaluation, how sure we are about what we see. So to conclude, we found evidence for a mechanism of identification and awareness in the frontoparietal network. We didn't find that the awareness was limited to the ventral stream. And finally, the superior temporal sulcus and the temporoparietal junction, instead of reflecting awareness activity, were more important for confidence evaluation or show we are about what we see. So to conclude, these two studies allowed us to introduce new paradigms that go beyond the seen-unseen approach, conscious-unconscious, that is often used in the cognitive neuroscientific studies of visual awareness. Each time we used one single task where the participant can choose one answer among 3 or more possible answers, and that allowed us to simultaneously evaluate several cognitive functions that are closely related: objective performance and identification, the subjective feeling of perceiving or awareness, and finally the confidence, certainty evaluation. So those functions usually go closely together, but using this gradual scale, we can start to try to dissociate them. And by doing so, we were able to see that visual awareness relies on a widely distributed network. So probably we are not going to find awareness in a few single isolated areas. So we have to go to the network level. And then that awareness is not just attended visual information. There is more components into this. So concerning this slide point, I think this understanding of visual information being selected by attention to enter visual awareness was an important aspect, but maybe is now limited, and we have to consider new theoretical framework. So I'm now going to introduce briefly how I currently think about consciousness. Consciousness is not a continuous stream of information. It's not a continuous flow, but it's made of discrete events which emerge from the meeting between ongoing processes and probes that trigger awareness of these processes. And the probes can be external signals or an internal questioning. So to give you more sense in what I mean by this model, as soon as you are awake and you open your eyes, you have some visual information coming to your brain. You have some spontaneous thought. You have some expectations on what is going to arrive in your visual environment. But all of this can be totally unconscious. It is only when a new probe arrives and crosses these ongoing cognitive processes that the consciousness can arrive. And the probe can be, for example, the onset of a new signal in the environment that may or may not match the signal expectations that you were maintaining unconsciously. It can be the decision to observe your own thoughts. Or it can be the fact that you are looking for something. The consciousness of looking at your room when you look for your key is very different than when you look for your room to see if it is harmonious or pretty, etc. So I think this is now the way we should, in the neuroscientific studies of conscious perception, be looking at consciousness, and we should design experiments to look into these different processes, this meeting between the ongoing cognitive processes and the probes. With that, I would like to thank my colleagues at the NIH, and I would like to thank you all for your attention.
Well, that was a very rich presentation, very rich 3 presentations. If I could invite, well, the other speakers didn't have to invite them, they're already marching up, as they should. If you could take a seat. I think we have 1 microphone to share between. Now, George moderated the question and answer session when I was on the panel, and he said, and I will repeat the same thing that he said, which is, please try to keep your questions concise, and please make sure they are questions. And we'll sort of alternate between rows. So we've got about half an hour for the panel discussion, maybe a touch less. So if we start here, you can address a question either to one speaker specifically or generally to the whole panel. Please.
Thanks. Here. For Dr. Vernet. This beautiful sculpture by Liechtenstein it made me think about the fact that schizophrenic patients have this problem that are unable to reconstruct. They would see that the house is concave and not convex. So do you think when your studies on Fmri, are you planning to do some studies in people that have these problems like schizophrenia, psychosis? And if you didn't do, what would you expect?
Thank you. Thank you for your question. I'm not at all a specialist about schizophrenia, but I indeed think that what I'm talking about, this dissociation between objective and subjective, can be really relevant for schizophrenia. As may hypothesize, but again, I'm not a specialist of schizophrenia, that we can maintain both this subjective feeling and some more objective knowledge about our own perception. So we can maintain both, and we can sometimes, not always, in the case of illusion, not always, but we can be able to metacognitively assess that our subjective feeling is maybe doesn't match with the reality. If we do the experiment, the Fmri experiment with schizophrenic patients, I might have the hypothesis that it would be more difficult to dissociate what is objective from what is subjective in these patients. Thank you.
Anyone else want to come? Okay. You have the next question.
Is this working? Yes. This question's for George. Actually, two parts. First part is the frontal feedback to the occipital areas and so on. Do you perceive that enhancement to be a result of increasing activity or suppression of activity in the occipital areas, for example? And have you measured whether that feedback is coming through the fasciculi or indirectly through the thalamocortical system, basal ganglia, and otherwise?
So great questions. First of all, I think you were asking about sort of this dominant activity.
Right, the frontal activity that sets off the awareness when the feedback comes.
So I don't know what the answer is. I think one possible neuroanatomic reason for the differences between rodent and human is just the degree of feedback connections structurally. But I think you bring up a great point, which is we're making an observation across the brain and making an implicit assumption that it's following a certain structural connectivity pathway. In fact, there might be cortical-subcortical dialogues going on along that pathway, so that's certainly a possibility. We try to address this, not across the whole brain, but specifically in primary sensory and primary motor cortex, which is right across the central sulcus. It represents the frontal-parietal divide, and we know that there's a polysynaptic structural cortical-cortical connection. So we're trying to dig down, but I don't have a full answer yet. And I also want to point that, I don't believe that there's some magical arrow that's going from the front of the brain to the back of the brain that's carrying consciousness particles. We think that it reflects a complicated process. It might be an epiphenomenon of the underlying network topology. I mean, it's still an interesting finding, but there are a lot of questions to answer.
Sure. Thank you.
Okay, thank you.
This is really a follow-up on the previous one. I understood you to suggest that the backward...
Yeah.
It's really hard to hear you, actually.
That the backward sweep...
Could you repeat your question from the beginning, please? I think we were having trouble listening to it.
Can you hear me now?
It's very muffled, actually. I think let's try to do something.
I understood, Professor Mashour, I understood you to say that an effect of the backward sweep was to either affect the intensity of what was happening at the back or the duration. Is there any other kind of effect as a result of the backward sweep? Is there any other kind of effect that you'd be willing to speculate happens?
So I want to be painfully honest and say I really have no idea what's going on. I mean, I don't know what it's doing. I don't know if it's an epiphenomenon. And I also want to point out that it really depends. I mean, I think there are newer data emerging suggesting that other conditions are going to give you a different kind of response. This is a resting state brain. I'm showing you snapshots. We're starting to have a better sense of the dynamics of that kind of connectivity and how it could change. I think in the context of the global neuronal workspace theory, this kind of reciprocal activity or recurrent activity is thought to amplify a signal or help sustain it over time because of the recurrent processing. But I don't really know the answer. I don't know if any of my colleagues do, but.
Thank you. Next question, please.
In an age where there's poor sleep hygiene, do you guys have any thoughts on the learning in a sleep-like state, especially if there's like poor sleep hygiene, like a TV on or something of that sort? Do you think that there's an impact there on, do you think there is learning that's possible during sleep, and do you guys feel like there's perturbance of that through like external sources?
Yeah, I suppose that your brain actually can learn, and we know a lot of time the long-term memory are reached to destinations in the sleep. So in that sense, yeah, your brain actually are active during the sleep cycle, and especially in the non-REM phase of it. And the question of whether if it's a novel information and you are learning the novel information under a state where you are not fully conscious, but your brain is processing the information nevertheless, then there may be some impairment. I don't know. It's really a very, very good question, but we don't really have a clear answer at this point.
Maybe I can just push you a little bit further on it, actually. It's me speaking. There's a difference here, an interesting distinction between a consolidation of memories and of learning processes during the day that we have very good evidence for happens especially during non-REM sleep, and the learning of new information during sleep, which was really the target of the studies, the beautiful studies that you described. And I guess I'm wondering that to just push on that question a bit further. What about sort of consolidation of memories during? How does that distinction play out in anesthesia, basically, between consolidation?
Yeah, that's actually an excellent question, and we only touched upon briefly or scratched the surface of it by comparing the learning of a mixture of odor with its component. And so the way of the experiment that we have designed is initially we want to look at the familiarity, not recollection of it. But it appears that the anesthetics effects during the initial learning did not only affect the individual component of the odor, but the association of the odor was blocked.
Right,
so during the second phase, it's actually learning the association as a new information. So it's being consolidated. So I cannot answer the question of consolidation at this stage, but apparently there's some difference in the familiarity and the recollection portion of the recall.
Okay, thank you. That's interesting. I think we're over to this side again. Next question, please.
So to use Jan's analogy, I'm still stuck in the nuts and bolts era, mainly because ultimately without that the structure falls apart, and that's where most of our interventions are right now. But the question is really for George. To follow up on some of these previous questions, this loss of anterior to posterior connectivity, does that have a discrete molecular correlate, or is that a system response to a global molecular issue?
I hate to profess my ignorance with every response, but I am in fact ignorant. I think it's an important question, and in fact, I've been trying to probe that. I mentioned Nick, who's a graduate student in pharmacology, but I've been connecting with a laboratory that really focuses on sodium channels, and we're trying to understand, is there a molecular basis for this? If we think about, what's responsible for, this information traversing from one area of the brain to another? Is there something on the ion channel level where if we model it that we could potentially account for this kind of activity? So I think it's a critically important question. I think if we're not bridging the gap between these different scales, we're not going to have a satisfactory explanation. So it is something that we're pursuing. Is it something that's rooted in some sort of molecular action, or is it just some kind of emergent phenomenon or epiphenomenon of a certain kind of network organization?
Next question, please.
Thank you. My question's for Dr. Mashour. I think I'm piggybacking after Dr. Eckenhoff here. So my question is, if I understood you correctly, is that anesthetics work by kind of snowing out these hubs to decrease their interaction? Is that correct?
That's a current hypothesis.
The current hypothesis.
The hubs might be preferentially affected.
Is there any rationale or reason why the anesthetic would target a hub versus a non-hub, and why would it target a hub that's related to consciousness versus something like heart function or some other process?
Right. So great question. Again, we have to figure out, is this a molecular event? So for example, there's a certain distribution of receptors in these hub areas. I suspect that's probably not going to be the answer. But one way I would think about it, and to use an analogy of Whisper Down the Lane, this childhood game, you have a certain decay of information as you're traversing these various messengers. And I think that it might be the case that a longer polysynaptic train is ultimately going to be more susceptible to the effect of anesthesia. So it might not be, for example, with this feed forward and feedback, that it's a particular directionality, but rather if you impair a lower order or monosynaptic or oligosynaptic process, maybe it will slow down. But when you have a hub, you're dependent on many different inputs. So it might be slowing down on one early level, but that hub function might just fall apart if not all the information is getting to the right place in the right time. So I'm inclined to think that that's probably going on. It seems like a more parsimonious explanation to me. I don't know about the receptor distribution per se, but my suspicion is that they're not going to be these clear hotspots and hubs where you have all of the molecular targets of a general anesthetic, but not in other parts of the brain, such as the primary sensory cortex.
Okay, thank you very much.
I just wanted to ask a question to all of the panel before we move on to the next two, which is, I think in many of the talks there was a quite interesting data about the role of the dorsolateral prefrontal cortex. And DLPFC has been the topic of a long-running dispute among scientists interested in consciousness. Is it the higher order thought seat? Is it necessary and sufficient? Is it a really key element of whatever network of brain regions is generating consciousness? Now, Maureen, in your talk, you showed that as the visibility ratings increased, you saw lots of activity in various brain regions, but not the DLPFC sort of kicking in. And then George, in your talk, you showed that cholinergic activation of the DLPFC was sufficient to reactivate conscious level, even while the animals were still breathing in anesthetic drugs. And I'm sure you said something about the DLPFC as well, but I can't remember exactly what it was. But I wonder if the panel could each just tell us what the role, if any, they think the dorsolateral prefrontal cortex has in consciousness. Maybe we'll start with Maureen and go down.
Thank you. Indeed, this was a surprise to me in my results to not find much more prefrontal activation. I think it depends on, again, what we call consciousness or awareness. So things might have a different role when it is the state of consciousness, like being awake versus being asleep, anesthetized. It can be different if we are thinking of higher order process, like reflecting on our own consciousness and using this information, like it's a little bit the access consciousness, what we use this information for. In the study I presented, it was more the subjective gradual feeling, which it's again, I found prefrontal activity, but maybe less so because it was more related to this subjective feeling, and other things were equalized, like the kind of response, the reaction time, etc., were taken into account in the models. So again, we all talk about consciousness, but slight differences in what we exactly put behind this word might explain this discrepancy we have across studies.
Thank you.
So I don't really have any data to speak to the distinction between phenomenal consciousness and access consciousness when it comes to the prefrontal cortex. I suspect that these early stages of anesthesia are disrupting access consciousness. I mean, I feel pretty comfortable suggesting that probably the disconnection, the consistent disconnection of the prefrontal cortex from the rest of the brain contributes to a loss of access consciousness. Like sleep, anesthesia is associated with dreaming. There are subjective events. The dream recall has a similar rate if you're interviewing somebody just after anesthesia as after sleep. So there is some residual phenomenal consciousness going on. In terms of the level of consciousness, just to be clear that and I'm glad you brought this up, because the area that we were probing was prelimbic prefrontal cortex in the rat, which is medial prefrontal cortex. And indeed, it changed the level of consciousness. I don't know if the content of consciousness changed. It could have been a zombie rat, so to speak. All we can measure is the observable of wakeful behavior. But again, it seems like, if not the clear locus for consciousness, that the kind of consciousness that we're experiencing right now that we can talk about and reflect upon, that we can attend to, that's associated with arousal, the prefrontal cortex does seem like it can have an influence on a number of those different key dimensions.
Thank you.
Yeah, well, so thinking of this, actually, this will also answer the previous question that we thought about this for a long time. And our theory, we believe that there's nothing in the brain of the connectivity that's deterministic for consciousness. But nevertheless, because of we know the density of the fibers in brain, different regions are different. So if you think about the probability of some anesthetics that come to suppress some of it, if you have a very high dense fibers, and if you have the same percentage of turning off that, the amount of fiber being turned off is much more than other regions. That's consistent with the idea of this hub. So you have in the brain, natural evolution already gave us these hubs of high density fibers, so that our theory basically says that there's no connection which is deterministic, but by the probability of access information, you will come to this phase transition where
so this is your percolation model, yes. Yeah. Okay, thank you. Now, my memory is terrible. Who's next?
I think
You're next. Great.
Hi. So, kind of branching off what you just asked, you mentioned, how each of our presenters mentioned DLPFC. I noticed the theme of, researching basically the neocortex and gray matter areas. I was curious if you think that there could have been, right? Let me rephrase this. If there would have been any influence, had you involved like transcranial stimulation into the mix? It's would it have affected the ability to learn or the ability to, I guess, I don't know, notice the differences in each of your experiments? In theory.
Well, I'll just comment briefly that, TMS is being used in terms of a perturbational approach to assessing consciousness, and I think that's been a robust approach, more rooted in integrated information theory and the breakdown of effective connectivity during sleep or general anesthesia. In fact, Dr. Bob Pierce, I think, is in the audience, who did some of that work. So I think that is important to bring up. Certainly the work that I was showing was not perturbational. We were looking at spontaneous activity, and I think there are probably differences if you're perturbing the system or engaging the brain in a task. I don't know if anybody else is.
Transcranial magnetic stimulation can be used in different ways, and one way of using it is to use rhythm to entrain brain oscillations at a particular frequencies. And we have previous studies where we have been doing so by stimulating the frontal eye field at beta frequencies. We were able to increase detection, so conscious perception of a briefly presented target again. So, and this increase of conscious perception with induction of oscillations was related to white matter connectivity between parietal and frontal areas. So I think this is a really relevant point to use TMS to probe causally this relationship between anatomy, oscillations, and conscious perception.
I'll just add something very quickly. In our lab as well, we've been using theta burst TMS, which again, it's a repeated stimulation that's supposed to induce a virtual lesion, supposed to deactivate a region temporarily. We were trying to assess the role of back to prefrontal cortex, dorsolateral prefrontal cortex, in metacognition. So in knowing that you know the stuff that Maureen was talking about as well. And we're now in a back and forth with Hakwan Lau's lab in UCLA, because they had evidence that this theta burst of prefrontal cortex produced a deficit specifically in metacognition, which you can associate with visual awareness, and then we just didn't find it. And it wasn't quite the same design, but it really was difficult it was a small effect at best. And so I think it's quite fragile. And of course, the other thing is you stimulate one region, it's going to have a lot of knock-on effects elsewhere. So you really don't know exactly what TMS is doing, even if you know where you're stimulating. At least that's one of the things we worry about.
Thank you.
Thank you. Question?
Yeah, this question is mostly for George. So I'm also interested in the front to back directional connectivity, the frontal parietal cortex. I was mostly wanted to ask about exactly what you just mentioned, that namely under some anesthetics, and ketamine in particular, people do report vivid dream experiences. So in the context of the Lee paper showing that this frontal parietal connectivity decreased just like other anesthetics, that from one perspective could be a problem. I mean, you could interpret that to show that indeed frontal parietal connectivity is not needed for conscious experience in the sense of phenomenal content of dreams and so forth. And we also have evidence showing the same things happening during REM sleep, that you get a decrease in front-to-back connectivity from frontal parietal cortex, but nevertheless people report vivid dreams too. So I'm just wondering how you think about that, and in the context of what you're just saying, whether you think it's just access consciousness or that.
Great question. I'm really glad you brought that up. It gives me an opportunity of following up on the perturbational complexity index to talk about a study by Sarasso et al. In Current Biology in 2015, where the PCI with ketamine really didn't go down that much, in contrast to the GABAergic anesthetics, non-REM sleep, pathologic states of unconsciousness. So this brings up another distinction of connected versus disconnected consciousness. Connected consciousness would be the kind of consciousness we're hopefully experiencing right now, where we're aware of the environment, versus disconnected consciousness, which might be mind wandering or a dream state or hallucination. So I guess my best guess right now is that there's something about this pattern of anterior to posterior connectivity that relates to connected consciousness, but might not really be capturing what's going on with disconnected consciousness. I mean, I think it's sort of related to access and phenomenal consciousness, but something that we talk about in the context of general anesthesia, and what we're potentially trying to do, and this discussed in the literature by Rob Sanders and Giulio Tononi and Stephen Lories and Jamie Slay, that really what we're trying to do with anesthesia is to suppress connected consciousness. Disconnected consciousness might be benign in the setting of a surgery. So thank you for bringing that up.
This question is for Dr. Vernet. Have you considered examining subjects who suffer from blindsight? It might be a way of teasing out aspect consciousness from phenomenal consciousness with your studies.
Yeah, it's a very good question. I did not record a participant with Blindsight, but I would be very happy to do so. The tricky question with Blindsight is that they maintain some level of objective performance while losing this subjective feeling. And one question I have in my mind relating to this question is, could these participants be trained to regain trust in their own objective performance so that they would gain an objective feeling? So a way to go back from the objective to the subjective. But this is an open question for me.
Thank you.
Thank you. We are drawing to the end, so we have, I think, 2 quick questions left, but I'm afraid nobody else stand up because we'll have to draw it to a close there. But I'm sure the speakers will be milling around afterwards if a burning question arises at the last minute.
Okay, this relates to Neil's question regarding dorsolateral prefrontal cortex. And I think it was George that said that some of the feedback comes from the medial prefrontal. My guess is that most, and even from one of your diagrams, George, but my guess is that most of the feedback in normal perception, normal visual perception and so on, is from ventrolateral prefrontal, and maybe even lateral occipital, OFC type of thing, rather than dorsolateral. Dorsolateral prefrontal, we tend to see as the big, boogeyman in our conference and so on. But dorsolateral is more involved in the control type of thing or executive type of thing, whereas normal perception probably involves more the ventrolateral prefrontal. Could you comment on that?
So first of all, just to be clear, when I talked about the medial prefrontal cortex, that related to where we were reverse dialyzing in norepinephrine or carbachol. So I wasn't suggesting that that was the origin of feedback projections per se. And also, when I was speaking about the dorsolateral prefrontal cortex, it was in the context of this recent article in Science where they recorded from that area and suggested that, in fact, these prefrontal cortical neurons were participating in visual experience. So again, I don't know. I wish I was more dogmatic. I'd be a lot more compelling in these Q&A sessions. But I think that's where the work needs to be done. I did show you some network data that just based on the neuroanatomical scaffold, just based on the topology, that you do see various nodes in frontal and prefrontal cortex appear to be, the phase leading nodes, and these areas in the posterior cortex tend to be phase lag. Of course, that's on a longer time scale. So further work needs to be done.
And last question, let's keep it tight, we're out of time.
It's to Marine and beautiful experiments. I really admire a lot. My questions concern the time scale of the experiments and the time scale of the imaging, and also something else, which is the movement of the eyes. And remembering Alexander Luria work on the examination of a painting and the effect of the frontal lobe, and all the different kind of meaning he can get through the exploration of the picture. I was wondering whether there is, since we have constantly saccades how do you fit with what you say is in one saccade you have the overall kind of process taking place or not?
I like your question a lot because I'm coming from the eye movements field. But now I'm doing experiments where I'm always asking my participants to fixate with the center of the screen, which is a bit frustrating for me. So we avoid some confounding of or contamination of brain signals, like if we do EEG or MRI. So the idea will be that by each saccade you move your attention spot and you collect a bit more of information so that you can then integrate into the percepts that you are building up. And I'm not so sure that I want to think about a representation that you have in your head and then bringing more and more information into it. But I think we need some kind of integration along the time so that each snapshot that we have at each, after each saccade can contribute to the building up of the conscious percept. I'm not so sure to understand the question about time. The first part of your question was about... We can talk about this later.
I think we should now, we're 5 minutes over time, so I think we can follow this up after the end of the session. But before we thank our speakers again, let me just remind you that the concurrent sessions, wherever they are, they're all over the place, they're concurrent. They start at 5 o'clock, and there is also a poster session which starts at 6:30 in the grand ballroom, and there will be booze. So with that, thanks to all our wonderful speakers for this afternoon.